everyone, my name is Tamal Ghosh and I am studying in IPGMR and SSKM Hospital, and I will be teaching you molecular biology. The topics discussed are DNA, its structure and properties, RNA, types and their properties, replication and repair, transcription, translation, regulation, mutation, and RDT and PCR. RDT stands for recombinant DNA technology and PCR stands for polymerase chain reaction. Okay. Now the main topic we are starting is the DNA. As the name suggests, it is deoxyribonucleic acid. Okay. It is a complex molecule. It is mainly the genetic component of all the organisms. I won't say all the organisms, but of maximum organisms. They have their genetic code. These genetic code are taken and the bodies are formed. So DNA is very important storehouse for all the specific genes present of the human, or I will say or every organism. Okay. It is a complex molecular structure which contains all the genetic data required for formation, growth and development of an organism. Okay. So this is DNA. Now the structure of DNA. The structure of DNA is mainly said by Watson and Crick in the year 1953. The main structure we are discussing is the B type of DNA. There are different types of DNA present in the in organisms. Some are A type DNA, some is B type DNA, C type DNA, D type DNA, and Z type DNA. Here the main DNA is the B type. Okay. The B type DNA is present in maximum organisms. And this DNA is discussed, this structure of DNA is discussed by Watson and Crick. In 1953 in their famous model. So we are discussing this structure now. Okay. So the structure is right-handed helix and each turn has 10 bases. Okay, there are two groups present. One is the minor group and one is the major group, and the length of each turn is 3.4 nanometer. Okay, so I am drawing the diagram here. This is their DNA. It has two ends, 5' and 3' end. Simultaneously, when you go here, one will be 3' and one will be 5'. Okay. Now this is a whole, I should use another color here. Okay. Now this is a whole turn. Okay. And this turn is 3.4 nanometer, has 10 base pairs present. It is a right-handed helix. It has two groups. One is the major group and another one is the minor group. Okay. It has complementary bases, that is 10 base pairs present. I am drawing this one by one here. This is 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. These 10 base pairs. Now the base pairs are made from the nitrogenous bases, that is adenine, guanine, cytosine and thymine. Okay. These are the 4 nitrogenous bases, and these groups are present for binding of the regulatory proteins. Means groups are present for binding of regulatory proteins. DNA is a vast molecular structure for its maintenance and regulation. Regulation in the sense the different processes taking place along with the maintenance of DNA. The regulatory proteins are very much required for its viability or maintenance. These both DNA strands are anti-parallel to each other. Here you can see that one is towards the 5' side and another one is the 3' side. They are both 5' side are not in same direction, or both 3' side are not in same direction. That means they are anti-parallel to each other, or I would say anti-parallel strands. So what are the importance we are getting here? That is, we are discussing the bDNA discussed by Watson and Crick. It has antiparallel strands, has 3.4 nanometer length of each turn, and has 10 base pairs, has major groove, has minor groove, and has 4 major bases, that is adenine, guanine, cytosine, and thymine. And they have right-handed helix. So these are the structure characteristics regarding the structure of DNA. Okay. Now the bonds which are forming here, we know that there are 2 types of bonds forming here. I will say 2 patterns of bonds regarding the 4 nitrogenous base. That is, 1, 2 hydrogen bonds between adenine and thymine. Okay. And another one is 3 hydrogen bond between cytosine and guanine. Here, the adenine and thymine, one is purine and one is pyrimidine. Okay. So what are pyrines? You know that in the metabolism nucleic acid, adenine purines are the adenine and guanine, and pyrimidine, but you know that they are thymine and what? Cytosine. The third one, that is uracil, is present in RNA. We will discuss it later. So pyrines, pyrimidine, that has adenine and guanine. Okay. Now there are some differences here. I will discuss it now. That is, first make this table. One is the nitrogenous bases, one is the nucleoside, and one is the nucleotide. While discussing it, I will discuss the backbone and the ladder structure of the DNA present. The nitrogenous base, as discussed earlier, are the adenine, guanine, cytosine, and the thymine. When the phosphate group joins with these nitrogenous bases, the nucleosides are formed. The nucleoside names are the adenosine, guanosine, cytidine. Here there is a trick that the nucleoside name is cytidine and not cytosine. Cytosine is the nitrogen base name here. Here, see that I am marking with another color here. So here is the cytidine, and the last one is the thymidine. And the nucleotide, I have said earlier one wrong thing. That is, nitrogenous base with pentose sugar forms a nucleoside, and along with that, if phosphate group is added, then the nucleotide will form. The nucleotide are the adenylate, guanylate, cytidylate, and last one, that is thymidylate. Okay. And this nucleotide finally forms the nucleic acid. Okay. So here, nitrogenous base plus pentose sugar plus the phosphate group forms the nucleotide. The nucleotide, different nucleotides, many, when added, forms the nucleic acid. Okay, nitrogenous base with pentose sugar with phosphate forms the nucleotide, and many nucleotide forms the nucleic acid. Okay. Now check this point and remember this point always: cytidine and cytosine. There should be no confusion here. Okay. Now I will discuss some rules as said by the Chargaff. These are named as Chargaff's rule. Okay. As I said earlier that adenine has 2 hydrogen bond with thymidine. This is a rule given by Chargaff. Another rule is that cytosine has 3 hydrogen with guanine. Okay. Now the third rule, as it seems very easy, that the number of purines is equal to number of pyrimidine. Okay. So if I see here that A plus C is equal to G plus T. Okay, as here we can see that always A is with T and C is with G. So if I add these two things, adenine and guanine, with thymine and cytosine, the number will be equal. Okay. sorry, I have wrong here, that is guanine, and this one will be cytosine. Now it is correct. I should make correct here, G and C. Okay. That means if I change it a little bit, A plus G along with C plus T is always equal to 1. This one is the third rule of Chargaff. Okay. And the last one is fourth one. The A plus T concentration and C plus G concentration is specific for each species, because some organisms have A, T concentration more than the C or G concentration. So this change can lead to different genetic material and different for different species. Okay. So remember this point: A plus T and C plus G specific for each species. Okay. I should delete this. Now, the basic characteristics and structures regarding the DNA are discussed here. Now, I will discuss about the next point is the RNA types and properties. So, what is RNA? RNA is the DNA is deoxyribo, and this is nucleic acid. Okay. Ribonucleic acid. The difference between the deoxyribo and the ribonucleic acid is mainly in the pentose sugar structure. I should draw the structure here, like this one is O, C, 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 and this one. Okay. In deoxyribose, here there is one H present at the second position. Okay, the first position has OH and H. Here the H and OH, H, H, and CH2OH present here. This is the structure of deoxyribose. And if I see the structure of ribose sugar, that is O, C, 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 same as that, but the difference here is that here OH present. Here is the H and here is the OH present, and remaining will be same H structure. This is ribose sugar. It is a main structural difference between the deoxyribose and the ribose. Now, RNA. RNA is mainly a genetic material, but not for humans or the maximum diversity of organisms. It is restricted to very limited amount of organisms, mainly in the viruses. As example, you know that the HIV has double ssRNA. Okay. Now, the RNA mainly has 4 nucleic nitrogenous base, that is adenine, guanine, cytosine, and in place of thymine it is uracil. Okay. In place of thymine. Okay. Generally RNA is present in single strand. Generally RNA present in single strand. As unlikely that DNA has DNA double strand, it is generally present in single strand. Okay. Now, the different types of RNA are types of RNA. Types include there are mainly 3 types. Okay. One is the rRNA, that is ribosomal RNA. Second one is the mRNA, that is messenger RNA. And the last one, that is the tRNA, that means transferring RNA or transporting RNA. The other different types of include the small cytoplasmic RNA, the small nucleolar RNA, microsomal RNA, etc., etc. Here are the main 3 RNAs. Okay. Now, as I am explaining that the RNA has uracil present. Uracil is the nitrogenous base, so the nucleoside will be uridine. Okay. And the nucleotide will be uridine monophosphate. Okay. or I would say, as the name here given, hemidilate, cytidilate, udidilate. Okay. Now they are synthesized by the help of an enzyme named as RNA polymerase. Okay. Formed by help of enzyme RNA polymerase. The RNA polymerase are generally of three types, that is 1, 2 and 3. Now these three have three different roles of forming the different types of RNA. Let me explain by a diagram. Like, take this as a DNA. Okay, now this is a DNA. The RNA polymerase 1 when acting, forms the rRNA. The rRNA can have different subtypes. The different subtypes may be like 5.8S. S is the Streptovirus constant. Okay, you already know that, like the 40S or the 50S RNA is present. Now the second one is the 28S and third one is 18S. If I take the second RNA polymerase 2, it will form the, first of all, primitive one, that is the snRNA, and finally it will modify it into mRNA after different kind of modifications. I will discuss it later, what are the different types of modifications. Okay, and the third one, that is the RNA polymerase 3. It will help to form the tRNA. Okay, you can remember this thing as 3 means T means tRNA. Okay, and polymerase 1 has all the 8 given numbers, that means 5.8S, 28S, 18S. So all the 8 ones are present in the RNA polymerase 1, and the second one, that is the RNA polymerase 2, have mRNA. Okay. Now I will discuss each and every RNA in little bit details. First one is the mRNA, or I should discuss first the rRNA. rRNA is the ribosomal RNA. It has the most concentration in any cell. Approximate 70 to 80% of all the RNA are the ribosomal RNA. All the RNAs are rRNA. They help in the formation of ribosomes. Helps in formation of ribosomes, as the name suggests. Okay. They mainly have the function of protein synthesis. As you all know that ribosomes take main function, that is the formation of the proteins. Protein synthesis. They also help in formation of the peptide bond. Formation of peptide bonds during peptide chain formation. Peptide chain. Okay. Now the ribosomes are different in case of prokaryotes or eukaryotes. If I take an organism that is the prokaryote one, or if I take an organism that is a eukaryote one, they have two different types of ribosomes present. The prokaryotic one has the 70S ribosomes. Means they have one larger subunit and one is the smaller subunit. Smaller. If I use the centrifuge machine and the Streptovirus unit, if I calculate it, one will have, the larger one will have the 50S and the smaller will have the 30S. Okay. The 50S has 34 types of proteins and the smaller one has approximately 21 proteins. Similarly, if I take the eukaryotic one, it has 80S ribosome. Also, it has larger subunit and the smaller one. The larger one is the 60S and the smaller one is the 40S. Don't confuse here with that the 60 and 40 is adding up to formation of the 100. Why 80 is present? It's not like that. The centrifuging unit is taken as the Streptovirus unit. If I take both the components together, the centrifuge unit is the 80S. But if I take each unit, each unit will have the 60S or the 40S one. Okay, that's why there is a difference. The 60S has generally 50 proteins and the smaller one has 30 proteins. So the important points here we have discussed already, that is 70 to 80% of all RNAs helps in formation of ribosomes, protein synthesis, formation of peptide bonds. And the prokaryote has the 70S ribosome and the eukaryote has 80S ribosomes. Now the second one, that is the mRNA. mRNA, that is messenger. What is the function of messenger RNA? Messenger RNA transfers the message from the DNA to the formation of the protein synthesis. So, transferring data from the DNA up to protein synthesis. Okay. They are up to 5 to 8% of total RNA. Their main function is to carry the information of DNA to ribosomes for protein synthesis. Understood? They are formed as precursor, that is the HNRNA. HN means heterogeneous nuclear RNA, and after some kind of post-transcriptional modifications, the mRNA is formed, that is preformed RNA, or that is the HNRNA, and HN stands for heterogeneous nuclear RNA, is formed into mRNA by post-transcriptional modifications. Okay. Now, the post-transcriptional modifications include there is formation of guanylate cap or the polyadenylate tail at the 3' site. Okay, I will discuss it here now. Some points, that is like, if I consider this is an HNRNA. Okay, there is the formation of one at the 5' site, and this is a 3' site. 5' site, there is capping by the 7-methyl guanosine triphosphate. Okay, this is helping in the formation of the cap, and at the 3' site there are many number of adenylate groups, that is polyadenylate tail. These are the post-transcriptional modifications. Okay, now here is the nitrogenous base is present and the formation of the mRNA. That is mRNA structure. Now, there is transport of the mRNA from nucleus to cytoplasm, and it is very much required because the main formation of mRNA taking place along with the help of DNA, and DNA is present in nucleus, but the ribosomes are present in the cytoplasm, so there is transferring of the mRNA from the nucleus to cytoplasm. This is helped by the capping, that is by the help of 7-methyl guanosine triphosphate. The capping helps to transfer mRNA from nucleus to cytoplasm. Okay, and they help to protect the 5' side from degeneration. Okay, you know that in bottles we need cap so that the water present does not get contagious or does not fall off, so it is protected. Same as the cap here, it is present, it protects the 5' side from any kind of degeneration or any peeling of the nuclear new nitrogenous bases. Same as the function of polyadenylate tail, it also protects the 3' side from any kind of degeneration. Okay, now the important points here, as I have discussed, are the transferring data from DNA to protein synthesis. 5 to 8% of all carry the information of DNA to ribosomes. Preformed RNA, that is the heterogeneous nuclear RNA, after post-transcriptional modifications forms the mRNA, and here the mRNA structure has capping, that is 7-methyl guanosine triphosphate and polyadenylate tail. Now the third one, that is the tRNA. Now the last one, that is the tRNA. The tRNA, that is the transferable RNA, otherwise they are known as soluble RNA or the adapter RNA. That means the transfer RNA or soluble RNA. It is mainly of 15 to 25% of all RNA. Now the structure is important. If I see as 2D structure, it looks like clover leaf. Clover leaf, you know that the structure is something like this. Clover leaf, something like this, the clover leaf structure. Okay. So if I see the structure of tRNA, I should add here the structure. Here, see this. This is the 2D structure. You can see that this something looks like this kind of structure, that is the clover leaf. Here one is the D loop, one is the anticodon loop, another one is the variable loop, and one is the T loop. Okay, now the D loop is, D means dihydrouracil arm, dihydrouracil. Okay, anticodon means the mRNA has the codon present, and along with it the complementary codons are present in the anticodon loop. The extra or the variable arm is present, and the last one, that is the T loop. T means thymidine pseudouridine cytosine loop. Okay, T means the thymidine loop. Okay. And this one is the 3D structure, and 3D structure is looking like inverted L shape. 2D structure is clover leaf. Okay. Now the examples include there are 20 types of tRNA. Because why 20 types of tRNA? We know that there are 20 types of different amino acids can be attached with it. They are helpful for the formation of the peptide bond. As the name suggests, tRNA, T means transfer. The tRNA helps to transfer the amino acids from different places to the ribosomes for the formation of the peptide bond. So what is the function? Transfer of the amino acids to ribosomes for formation of peptide bond. 20 types, generally 20 types of tRNA. Main functions, the transfer of amino acid to ribosomes. Okay. The main points I am pointing out here are the 15 to 25%. 2D structure is the clover leaf, also explained here. The 3D structure is inverted L shape. There are 4 loops present in tRNA, that is the D loop, anticodon loop, variable loop, and the T loop. And this end is known as the acceptor arm. The acceptor point, that is the CCA, point here, the acceptor arm. The acceptor arm present in here, in this point. Okay, I am giving a star mark here, so you will understand. The acceptor arm for attachment of amino acid with tRNA. Okay, now how this process takes place, I will discuss it later. Okay, the important points here: the transfer of amino acid to ribosomes, mainly 20 types of tRNA, and the acceptor arm for attachment of amino acid. Okay, now the next topic is the replication, as explained here already. The replication and the repair. Okay. And repair. So what is replication? Replication is basically the replica formation of the preformed DNA. Okay, we need replication so that the new cells are formed and the new bodies are formed from these kind of cells, and the new tissue forming will be helpful for the next generations or the repairment of the already damaged tissues. So new DNA is always required for the new cell, and the replication is very much required for this process. So In the replication there are different points to be remembered. The first point is the semi-conservative nature of the DNA. What is semi-conservative? That means when the DNA is replicating, the original one is present along with a new strand is also present. I will explain it. First I should write this point, that is semi-conservative. And next point is that it has an origin site and proceeds the replication process in both direction. Means if this is an DNA, it will break down into two parts in the origin site and the replication will take place in both the sides. Okay. Means bidirectional. So the process takes place bidirectionally. Okay. Now the synthesis generally occurs in the 5' to 3' side. Process occurs towards 5' to 3' side. Okay. Now this is sometimes degraded by nucleases. Degraded by the nuclease enzyme. If some degradation doesn't take place, the origin site will not be opened, and if the origin site will not be opened, the replication process will not take place. I will explain it with a diagram. Nuclease enzyme. Okay. It has high degree of accuracy. If accuracy is low, then there will be mutations, and mutations will be lead to altered protein formation or altered function of the cell. So it can be lethal also. High degree of accuracy. This is very important point, accuracy. It requires different enzymes. The enzymes include the DNA polymerase, helicase, topoisomerase, and etc. etc. Require many enzymes. Okay. Now I should show you a diagram here. See. Now see this diagram. This is the whole process of replication. Replication process. Okay. Now here we can see the different enzymes, that is the topoisomerase, helicase, DNA primase, DNA polymerase, and two different types of DNA polymerases. First I should say that there are different kind of DNA polymerases in eukaryotes. If I take the prokaryotes and eukaryotes, the DNA polymerases are somewhat little bit different. First, if I take the prokaryotes, the DNA polymerases are the DNA polymerase 1, 2, and 3. Okay, but in eukaryotes, there are no DNA polymerase 1 or 2, but the DNA polymerase 3 is present. This DNA polymerase 3 has 5 subtypes. Okay, the 5 subtypes include the 2, 3, 4, and 5. 5 subtypes include the alpha, the beta 1, gamma, epsilon, and delta. Okay, each one has their own function. The alpha has the function of synthesis of short DNA strands. Okay, the beta has the function of DNA repair. The gamma has mitochondrial DNA synthesis. The mitochondria has their own genetic material. You know that for its replication, the gamma is required. Mitochondrial DNA formation. Okay. The epsilon has the function of leading strand synthesis. Now, what is leading strand and what is lagging strand? I will discuss it along with the diagram. Epsilon leading strand synthesis. And the last one, that is delta, is lagging strand synthesis. Okay. Now see this diagram here. This is a DNA and it is being replicated. Here we can see the DNA polymerase enzyme present. Okay, this is helping in the replication process. Here we can see the DNA polymerase again. This is helping in the replication process. Now RNA primer is also present. This RNA primer is very much required for the founding stone of the replication process. Means foundation stone for the replication. Without this primer, the replication will not take place. Okay. And with the help of DNA primase, the RNA primer is formed and changed to DNA so that the replication will take place. Actually, it is taking place in the DNA, na? So a DNA primase is required. Now helicase. Helicase helps in the uncoiling of the DNA. Okay, helicase is here. This helicase helps in the uncoiling of the DNA. And the topoisomerase uncoils the DNA. Means there is, when there is helicase acting, there is supercoiling in the behind part. This is the behind part, and here the supercoiling is taking place. Supercoiling. So when there is supercoiling taking place, it will be difficult for the helicase to break the DNA, break the double-stranded DNA. So for its easier action, the topoisomerase is acting here. So the different enzymes are, as I already explained here, the DNA polymerase. The others are the helicase. I should use another color. Helicase breaks the double-stranded DNA. Topoisomerase uncoils the supercoiling. Others are the RNA primase, helps in formation of RNA primer, DNA primase, formation of DNA primer, and ligase. Without ligase, the finally attachments of the DNA will not take place. Okay, so this is a very important enzyme also. Adhesion of the strands. Okay. Now here I can see other points. You see here the DNA ligase present, and here are the Okazaki fragment. Okazaki fragments takes place in the lagging strand formation. Okay, as I said earlier, the replication process takes place from 5' to 3' side. Okay, so if I see here the 5' to the, here see that the 5' to 3' side, it is leading strand. Here, as I am explaining with a black color marking, I should use another color. Here, see, this is the leading strand, and it is forming from the 5' to the 3' side. So it is a leading strand. But here we can see that it is forming between 3' to 5'. So it is the opposite of the leading strand. So it is the lagging strand. And while formation of lagging strand, there is a problem. You know that I would say that against the rule of nature, the strand is forming. So small fragments will be forming. These small fragments are known as the Okazaki fragments. Okay. Now this is an Okazaki fragment. This is another Okazaki fragment, and this way it is forming. Okay. So what is Okazaki fragment? So what is Okazaki fragment? It is small segments of DNA formed during replication complementary to the lagging strand. These small segments of DNA are later attached together by the help of DNA ligase, as I said earlier. So if I show you a diagram here, that I should use another color. This is a replicating fork. You see that this something looks like a fork. That's why it is also called as the replicating fork. Replicating fork, and this is the 5' to 3' side, and this one is 3' to 5' side. Now if I take the enzyme of the Okazaki fragments always forms against the rule. That means from formation of 3' to 5'. If I take this one 3' and this one as the 5', so the lagging strand will be formed something like this. This one is an Okazaki fragment one, this one is another one, this one is another one, and it goes on. But the leading strand is always forming between the 5' to 3' side. So the new forming 5' to 3' is something like this. This is the 5', that is the 3', and the small ones are the forming like this is the 5', this is the 3', this is again 5', this is again 3'. Okay, so this one is the continuous strand, and these are the Okazaki fragments. Okay, I hope you understood this point. And as I see here, this is a 3' to 5' one, so this is the lagging strand, and this one is the leading strand. Okay, now semi-conservative, as I was explaining. So semi-conservative model was also given by Watson and Crick. Okay. So what is semi-conservative? Let us think that this is a DNA. Understood? 5' to 3' and 5' to 3'. Now if the replication takes place, as I explained earlier, something like this, then the new strand that is forming will be half of the old strand and half of the new strand. I am giving you a diagram here. Let us think that this is one strand forming, this is one strand getting divided, and this is the another strand. Okay, now this is 5' to 3' and simultaneously this is 5' to 3'. Now if the replication takes place, then the new strand formed will be something like this. This is 5' to 3', and the green one is the new one, that is something like this. Again, I am drawing here. This is the one, and this is another one. So here we can see that 5' to 3' is forming, and simultaneously 5' to 3' is forming. Again here, this is 3', this is 5', and this is 3', this is 5'. So here we can see the old one is also present. Old one is also present, and the newly formed strand is also present. So this is semi-conservative, not fully conservative, and not newly formed. Half of it is newly formed, thus semi-conservative nature. Okay, now here I missed out a point, that there are different small proteins present. These are known as single stranded binding proteins, that is SSBs. These proteins are very much required for the maintenance of the stability during the replication process. Here there is dynamic changes in the action of the replication. If these proteins are not present, the DNA will be disaltered, or there will be some kind of damage. For its regulation, the SSBs are present. Okay. Now here I explained the semi-conservative nature. Okay. Now I should point out the important points here, that in the replication process, there are, in prokaryotes, there are 3 types of DNA polymerase. In eukaryotes, the DNA polymerase is present only, DNA polymerase 3 is present only, and it has 5 subtypes, as I explained here: alpha, beta, gamma, epsilon, and delta. The different enzymes acting here are the helicase, topoisomerase, RNA primase, DNA primase, and DNA ligase. Now what is Okazaki fragments? Okazaki fragments are the small segments of DNA formed during replication, complementary to the lagging strand. And what is lagging strand and leading strand, as I explained here? Okay, now this is the semi-conservative nature. Now if I take the replication process of prokaryotes and eukaryotes, they are little bit somewhat different. So I should explain one with the help of a flow chart, I would say. This one is taken as the prokaryote replication, and this one is eukaryote replication. Okay. Now in replication of prokaryote, there is one origin site for the replication in the whole DNA. This is known as the ORI-C site. That means origin site for the replication, that is ORI-C site. Now there is unwinding of the DNA, by the help of nucleases. There will be formation of DNA bubble, or replication bubble, like something like this at the OD site. This is the replication bubble. Now this strand breaks here, and if the strand breaks here, then the formation of the replication fork, something like this, replication fork is formed. Okay. Now by the help of helicase and acting bidirectionally, there is progressive unwinding of the DNA taking place, and the help of topoisomerase, the supercoils are being dismantled. Helicase and the topoisomerase action taking place. Helicase is unwinding the whole DNA strand, and the topoisomerase is removing the supercoil. Now, by the help of SSBs, that is the single-stranded DNA binding proteins, the strands are in stability. They also help in the breakdown process of the strands, so that if nobody holds the DNA in its proper position or in the shape, the breaking by the help of helicase or the unwinding by the help of the helicase will not take place. Okay. Now, I am getting here 3 parts after this breakdown. One is the leading strand, one is the lagging strand. Okay, I should use another color here because it will be easier for you to judge these points. Okay. Now, the leading strand and the lagging strand. The leading strand helps, it is in the 5' to 3' direction, and this is also 5' to 3' direction, but the changes here is the continuous synthesis of the DNA takes place. As I said earlier, here there is continuous synthesis, and here is the discontinuous. Here I should give it here like this one. Here no Okazaki fragment. Here Okazaki fragments are formed. It is towards the replication fork. And this is away from the replication fork. But these are the important points here you should understand. Now, by the help of DNA polymerase 1, 2 and 3 in the prokaryotes, the whole replication takes place. Now, the DNA polymerase 1 has the action of removal of RNA primer and the DNA synthesis. The DNA polymerase 2 has the proofreading action, and the DNA polymerase 3 has the whole length of the DNA formation. Okay. Now, here is the eukaryotic replication. Eukaryotic replication is somewhat different from the prokaryotic one. Here there are different origin sites present. There are multiple origin sites. Okay. So, multiple unwinding, then multiple bubbles, replication bubbles. Okay. Some diagram I should make here like this one, this and it goes on. Okay, multiple replicating bubbles. Now again, here is the presence of the helicase and the topoisomerase. They are helping in the replication process, as I said earlier. Now similarly here, replication fork, and then the action of helicase and topoisomerase. Yeah. Okay. Now here some are different action of the DNA polymerase, like the DNA polymerase alpha. As I said earlier, it is helping in the formation of short stretch of DNA. So after the formation of here, after replication fork, as I said, the RNA primer is the main foundation in the DNA replication process. So RNA primer is formed by the help of RNA primase, as I said earlier. After that RNA primer, the small DNA strands, that is the main foundation DNA strands, are formed by the help of alpha polymerase. Means alpha DNA polymerase helps in the formation of short DNA strands. Here there are a factor known as the replication factor C. This is helpful for the replication process. There are different factors present here, but the most important you should remember, that is the replication factor C. Now, replication factor C is present. After this action of the alpha DNA, the action of epsilon and the gamma DNA polymerase takes place. Now you know that the epsilon 1 takes place on the leading strand and the delta 1 takes place in the lagging strand. Okay. Now the whole polymerization of the nucleotides takes place. Now polymerization of nucleotides takes place. Now finally a molecule, that is, I would say enzyme, that is a ribonuclease H. This ribonuclease H helps in the removal of RNA primer, because after the DNA replication the RNA primer is not required, it has to be removed, or else it will do something wrong to the DNA replication process. So the ribonuclease H present for the RNA primer removal. Okay. And finally the action of DNA ligase for all attachments of the Okazaki fragments or the leading strand, any replication or the repairment is taking place, etc., etc. So these are the main two differences in the prokaryotic replication and the eukaryotic one. You should remember these points. Hopefully you understood these points. Now the next topic is the repair. After the replication, there can be different alterations or there can be different mistakes in the replication process. This needs to be repaired, or else it will lead to any kind of mutation. So the next topic is the repair. Now the DNA damage can take place in the physiological manner during the process of replication. So DNA damage can be during replication, or there can be some agents present that can lead to DNA damage. There can be presence of free radicals that are too much harmful for the DNA. There can be some physical agents like the UV rays, X-rays, gamma rays that are also harmful for our body. UV rays, X-rays, etc. Or there can be some chemical agents like the nicotine, hydrazine, ozone. They are very much potent for the damage of the DNA. Ozone, nicotine, hydrazine, etc. Now, what are the different types of DNA damage? Types of DNA damage. So what are the types of DNA damage? Remember the mnemonic STCC. Okay. Now, what is the S? S stands for the single base alteration. Single base alteration. So what is single base alteration? There is changing in one base, one nitrogenous base. As the name suggests, there is a single base alteration. One base change. If there is change in the one base, then there will be in the process of the DNA damage. Like if I take an example, in place of cytosine, there can be accidental presence of the uracil, or in the uric acid metabolism you also know that there is presence of the xanthine. So if there can be any modifications or there can be DNA damage, adenine can change to the hypoxanthine. Examples, if I take like adenine to hypoxanthine, like, or the examples include the alkylating agents that helps in the single base alteration. Okay. Or there can be change between the addition or insertion of any nucleotide. Means there can be insertion of any nucleotide in a single nucleotide, I would say. Okay. So as the name says, single base alteration. Now the next one is the two base alteration. Two base alteration, as the T suggests, means two base alteration. Here there is generally formation of the TT dimer. Okay. Formation of TT dimer. Now the point arises, what is TT dimer? Okay. So T means thymidine dimers. These dimers are produced when there are adjacent thymidine residues are present, and due to some kind of DNA damaging products like that of UV radiation or the X radiations, there can be some covalent linkage. Thus, there is formation of the thymidine dimer. So, if I explain it, that is thymidine dimer, and thymidine dimer means two adjacent thymidine residues are bonded covalently after exposure to UV rays or some kind of X-rays. This can lead to two base changes, and these two base changes can lead to some kind of frame shift mutation. I will discuss about the frame shift mutation in the mutation topic. Okay, just right now we remember this name. So this is the thymidine dimer, two base alteration. Now, as I said here, STCC means the third one, that is a C, means chain break. As the name suggests, chain breaks. So the whole strand is forming, and suddenly there is some kind of breaking of the DNA part. It means if there is some kind of DNA breaking, so the chain is broken. So chain break. This chain break can take place by some kind of alkylating agents or the ionizing radiations or the UV rays or the X-rays or something, or there can be some kind of radioactive disintegration. So chain breaks means breaking of the strand can occur due to alkylating agents or some kind of radiations. Okay, now the last one, that is cross linkage. Now, as the name suggests, there is linkage between different nitrogenous bases. These nitrogenous bases can be present in the same strand or the different strands. So according to the name, stands linkage between different nitrogenous bases which can be present in same strand or in different strand. Okay, so this is the cross linkage. So what are the four types of DNA damage? That is single base alteration, two base alteration, chain break, and the cross link. Now there are different DNA repair mechanisms, okay, but mainly there are 5 types. I will discuss it one by one. So DNA repair processes. Okay, first one is the base excision repair. The second one is the direct repair. Third one is mismatch repair. Fourth one is the nucleotide excision repair, and another one is the double strand break repair. Now these are the important repair processes. Now I will discuss it one by one. First one is the base excision repair. As the name is giving you a hint, that is base excision repair. The one nitrogenous base which is forming the deformity or which is altering the action of the DNA is excised out, and the correct nitrogenous base is attached. So repairs the DNA by excision of the nitrogenous base, I should say wrong one, and replace it with the correct nitrogenous base. Okay. So like if I give an example, in case of, as I explained earlier, there can be some spontaneous deamination leading to the formation of cytosine to uracil or something like that. Now another point here is the no nucleotide is removed. Here remember this point, because in latter part there is one nucleotide removal. No nucleotide removed. Okay. Now if I give a diagram here as example, think this is a DNA strand. Okay. you know that in place G always C is present. But if there is some spontaneous deamination, or I would say some kind of DNA replication process problem, there is change of C to U, means cytosine is absent. In place of cytosine, uracil is present. So this is the wrong one. We have to correct it. So while correcting it, only the uracil is removed and whole lot of nucleotide is not removed. Okay, so in G, and here it is removed at this point, and use another color. This point it is removed. Now here same again, this one G is present. Here there is one breakage here, or else the nitrogen base will not be removed. Now the new one comes here, C comes here, and with the help of DNA polymerase again, G and C, help of DNA polymerase, and another one, that is ligase, because here a breakdown is done. The newly formed, repaired DNA is formed. This is the base excitation repair. Now the next one is the direct repair. As the name suggests, in the direct repair there is repairment of the whole lot of that, I would say whole lot of that DNA. Now the next one is direct repair. So as I was explaining, the direct repair in this process, there is no breaking down or no exchange of one nitrogen space to another one. But here we can see that there are different molecules and enzymes taking place, or the chemical agents taking place, that alters the changes in the molecular structure of the damaged DNA part, so that the newly formed DNA is seen. Like they act on the damaged nucleotides or nucleosides, so that converting in original structure. Let's say, write it here, direct repair. So this acts on directly damaged nucleotides and changing the damaged one to new one without any breaking or exchange of the nucleotide. If I give you an example, like you have a toy. I am giving you as a real life example that you have a toy and it is somewhat damaged. Well, you can do two things. Like one, you can change the damaged part of that toy with a new one, and another one you can do is the damaged part you can repair it yourself and made it correct, and you can play with that toy. So there are two different processes. So if I compare it with the base excision repair, the base excision repair is said as that you change that damaged toy part with new one. So this is base excision. But the direct repair means that you repaired the damaged toy part and you set the original one which was previously given after repairing it. So this is the direct repair. Now the third one is the mismatch repair. I guess it is mismatch repair. Yes. Okay. So what is mismatch repair? Mismatch repair is the repair of the mismatched base present in the DNA. Repair of mismatched nitrogenous bases. Okay. Like we know that, for example, along with A with double hydrogen bond, the T is present, or with C triple hydrogen bond, G is present. But if in DNA, if I see that sometimes like this A and T is present, but if any kind of DNA damage taking place and in place of A there I see C is present, so this is wrong now. So if I want to correct it, what I will do? I will do the repair in the mismatched N bases. So I will excise that part, make a A and make this part a hollow point, something like this, a hollow point, by the help of exonuclease. By the help of exonuclease, I removed that point. Okay, I removed that damaged sequence. And the new DNA sequence I am adding here, that is the T. Okay. The T is same by the help of DNA polymerase. Okay. And after that, DNA ligase is also taking place, because here there is breaking down by the help of exonuclease. So after exonuclease action and DNA polymerase action, DNA ligase action will also take place, and the corrected base is added. So now the next one is the mismatch repair done. Now the nucleotide excision repair. As the name suggests, nucleotide, the whole nucleotide is excised and then there is repairment taking place. In the nucleotide excision repair, I said the TT dimer. Nucleotide excision repair. As I said, the TT dimer present in the adjacent one. So if I draw a diagram here and here, TT dimer present here. Okay. This is excised. Okay. After the recognition, it is excised, and there is a hollow point formation. Hollow point. Then again, by the help of DNA polymerase and DNA ligase, the corrected ones are attached here. Okay. And new DNA formed. Okay. Now the last one, that is fifth one, double stranded break repair. Okay. Now here both the strands have been damaged and they are repaired. Okay, like if I show it with the diagram, this is our DNA and both these strands are damaged at this point, maybe I would say. So what will happen? These damaged strands are excised. Now after excision, there is action of KU proteins for the activation of DNA dependent protein kinase. These KU proteins helps in recognition of the break site. Okay. After that, this excision, there is approximation of DNA strands. Means approximation of DNA strand. Approximation means the nearing points of this DNA strands take place, so that the repairing could be done much more easily. Approximation means closeness. This closeness is done here. Now the unwinding takes place. Unwinding takes place, and the repair is taking place. Repair takes place. Okay. After this repair, by the action of DNA polymerase and DNA ligase, finally the formation of the new DNA occurs. Like this. Formation of new DNA. So these are the main repairing methods or the processes. Now here always remember a point, that is in this DNA repair there are basically 4 steps. Mainly 4 steps take place in DNA repair. That is, first, recognition of the damaged site. Second one is removal of the damaged site regarding the requirement, whether it is nucleotide or the whole base pair or the strand, etc. Removal. Third one is synthesis of the new strands or the nitrogenous base or the nucleotide. The last one is attachment or adhering action by the ligase, right? That is ligation. These are the main 4 steps in the repairing process. Now the next topic is transcription. So what is transcription? Transcription is the synthesis of RNA from DNA. Synthesis of RNA from DNA. Okay. it has mainly 3 steps, that is the initiation, elongation and termination. 3 steps: initiation, elongation and finally termination. Okay. Now here is 2 types of theories. One is the central dogma of molecular biology and one is the reverse dogma. The central dogma of molecular biology says that always and always that from DNA by the process of transcription RNA is formed, and from RNA by the process of translation the proteins are formed. So central dogma is the from DNA always RNA formed, and from RNA proteins are formed. From RNA by the process of transcription, which we are going to discuss, and here translation. Okay. And the reverse dogma says that from RNA DNA can also form. This reverse dogma is seen in HIV by the help of the enzyme reverse transcriptase. Okay. Now, this RNA formation takes place by the help of enzyme RNA polymerase. RNA polymerase. This is a very important enzyme without which the RNA will not be formed. Okay. Now, RNA polymerase has a factor known as sigma factor. Sigma factor. Okay. This sigma factor helps in finding the promoter region. Now, what is promoter region? I will say in the next one. Like, there are different words which you have to understand while understanding the transcription process. The some points are the promoter, coding strand, template strand, upstream and downstream. Okay. These are the different points you have to understand for understanding of the transcription process. So what is promoter? And sorry, I have missed one point, that is the terminator. Okay. So now what is promoter? Promoter is the coding strand of the DNA where the RNA polymerase binds. Coding strand where the RNA polymerase binds. Okay. Now what is coding strand? Coding strand is the opposite strand of the template strand where the nucleotide sequence is that of the new RNA formed, except the in place of thymine the uracil is formed. So I should first explain the template strand. So what is template strand? This strand from where the RNA is synthesized. Using this strand RNA synthesized. This is opposite of template strand, same as of RNA, except in place of thymine, in place of uracil. Okay. Now what is upstream? The DNA sequence before promoter and their sequence after the promoter site. And the DNA sequence up to which the RNA formation takes place. Okay, so these are some of the points you have to remember it. Okay. So if I take an example here, like this one is the DNA strand. Okay, and and this one is the promoter site. Promoter site. So before this, I have missed one part, that if you think that this is the 5 dash, this is the 3 dash, this is the 3 dash, and this is the 5 dash. So the template strand will be always formation. If I take the RNA forming in this way, 5' to 3', so this will be the template, and this is the coding strand. Now you might think why this name is given. Template is given from which the printing of the RNA is taking place, and coding strand is given name as the RNA coding can be done by the help of this DNA strand. If as they are same in composition, na? Just only difference is in place of thymidine there is uracil present, or else everything is same. So the coding of the RNA can be done by this coding strand. Thus the name coding is present. So this is the upstream, and this one is the downstream, and the RNA is forming here. Now, these consequences in promoter. This promoter helps to bind the RNA polymerase. Now there are different specific sequences present in the upstream region which helps in the transcription process. One example is the TAAT box. Okay, like I should draw it here, like 1 2 3 4 5 6. So this is a box. Now this is known as TAAT box. This helps in the proper binding of the RNA polymerase. They are present in the upstream region of the DNA sequence. Now the 3 steps for the transcription are the initiation. Now initiation of the RNA formation. So in this step there is attachment of RNA polymerase to the promoter region. Okay. Attachment of RNA polymerase to promoter sequence. This RNA polymerase has sigma factor that helps to identify the promoter region. So sigma factor helps to find the promoter region. Okay. Also helps in binding. Now after this binding there is a opening of the DNA strands, or else opening, I would say unwinding of the DNA strand, or else how will the RNA polymerization will take place. So if I show it with a diagram, this one is the DNA sequence and this one is the RNA polymerase, and in this direction the RNA polymerization is taking place. So there will be unwinding of the RNA site, and here the RNA polymerase is active. Now as it goes to this side, slowly the new RNA is formed. Okay, so I will draw a diagram here again. It goes here, something like this, and the new RNA is forming here. Okay, new RNA forming, and it goes towards this direction. Okay, now here we can see that there is no need of primer, and first the short stretch of RNA is formed. Here no need of primer, direct formation of the RNA takes place, and first short stretch is formed. Okay, now after the initiation the promoter is getting backed, means they after the promoter site, this is the P site, they left the promoter site at the backward point and it goes forward and forwards towards the replicating direction. Okay, so there is shifting of the RNA polymerase while forming the new RNA strand. These are the main steps taking place in the initiation. So another point is the from promoter side the polymerase shifts and the formation of new RNA strand takes place. Okay. Now the next point is the elongation. Okay. So in elongation, there is the RNA strand will grow and grow, and the whole new RNA strand is formed. Okay. There is a formation of 3' to 5' phosphodiester linkage. There is no still now there is no proofreading by the RNA polymerase. Okay. And I should write this point. The chain of RNA will elongate. Still now no proofreading taking place. Understood? And nucleotides are getting attached to the 3' end of RNA strand. Now finally there is termination. So termination. Now termination occurs by the two processes. One is the intrinsic, means in DNA there are some sequences which help to terminate the RNA polymerase activity. Okay, and another one is the extrinsic, means there are some factors which help to terminate the transcription process. So in intrinsic termination there can be some signals like polyuracil in RNA. Polyuracil means after the formation of mRNA there are different U and it goes on, and it helps in the ending the transcription process. And also the GC terminal, it's like more or like hairpin structure. Okay, these two are mainly taking place in the intrinsic factors for termination. If I give you a diagram like this is the DNA, and if the RNA is forming here and here and here like this, finally there is a hairpin like formation, and after that the poly U formation, means this is the 5', this is the 3', this is again 3', and this is 5'. So here GC containing the units GC, and it goes on, and here there are polyuridine residues. So these are the intrinsic factor for the termination. Now extrinsic factor means there is presence of rho factor. Okay, rho factor present helps to terminate the transcription process. Okay, now this requires ATP for stopping. Okay, so the three steps are the initiation, elongation, and termination. So what are the important points we should point out in the transcription are synthesis of RNA from DNA in three steps, that is initiation, elongation, and termination. Now there are two types of dogma, one is the central dogma and one is the reverse dogma. Central dogma says that from DNA there is formation of RNA by the process of transcription, and from RNA there is formation of proteins by the process of translation. In reverse dogma there is formation of DNA from RNA. Now RNA polymerase has sigma factor that helps in the finding of the promoter region. Now the some important points are the promoter. What is the promoter region? What is the promoter region? What is the coding strand? What is the template strand? What is upstream? What is downstream? And what is terminator? Okay. Now there is a TAAT box. This helps in the proper binding of the RNA polymerase. Okay. They are generally untranslated regions, means they untranscripted regions. They are present in the upstream part of the DNA sequence. This promoter helps in binding of RNA polymerase. Now in initiation there is sigma factor that helps to find promoter and in binding. Now from promotion side there is shifts and the formation of RNA strand takes place. In elongation the nucleotides are getting attached to the 3' end of the RNA strand. The chain of RNA will elongate and the other points will take place. Now the proofreading means there is no correction of the RNA takes place in this point. Now in the termination there are two types. There can be intrinsic one, there can be extrinsic one. In intrinsic one there is polyuracil residues and GC hairpin like terminal. Okay, sorry, there was a mistake. Okay. Now in extrinsic factor there is rho factor which helps to terminate in the transcription process with the help of the ATP. Now the inhibitors of the transcription. We know that this is the process for the prokaryotic transcription and if the inhibitors are required as antibiotics for the formation of new microbes in the body, so the inhibitors we have to know so that we can know the action of the antibiotics in prokaryotic transcription. One is the example of rifampicin. I hope you have heard this name before. And another one is actinomycin. Okay, both are used as antibiotics. Okay, the rifampicin is mainly used in tuberculosis treatment. Okay. So these two important points are the rifampicin and actinomycin. Now the transcription in eukaryotes. In eukaryotes there are different steps. I won't say different steps. There is little bit of different process than that of the prokaryotic one. Okay. Yeah, here it is same definition in transcription. This is the process of synthesis of RNA from the DNA. Now here is the twist that I have explained earlier that in eukaryotes, sorry, right here the first, in eukaryotes there are three types of RNA polymerase. Okay, this RNA polymerase 1 forms the rRNA. The RNA polymerase 2 forms the mRNA, and the RNA polymerase 3 forms the tRNA. I have said this earlier also, so I hope that you should remember these points. Okay, now here is also three steps, that is initiation, elongation and termination. Okay, so first one is the initiation. In initiation there is same promoters region where the RNA polymerase will bind and the finally transcription will take place. If I use this, so promoter, this one is the upstream, this one is the downstream. Okay, if I take this as 5' to 3' and this 3' to 5'. Okay. Now before the promoter region, in the upstream region, same as I said TAAT box, there is also box known as, I should point it out with this, that is the TAAA box. This also helps in the proper binding of the RNA polymerase in the promoter region. Okay. And this one is the coding strand, and this one is the template strand. Now don't get confused. While I was saying that the RNA formation takes place in the 5' to 3' site, that doesn't mean I am saying that the 5' to 3' site of the DNA. Okay, the 3' to 5' site is the template strand, because we have to form the RNA in 5' to 3', not taking the 5' to 3'. We have to form the RNA strand to 5' to 3'. So complementary strand, that is 3' to 5' is the template strand. You have to remember this point very much. Now this box is present in 25 base pairs before, in the, let's say 25 base pairs ahead in upstream. Means if I take this promoter base pair as the 0, so if I go to the upstream side 25 base pairs before, then I will get the TAAA box. Okay, now there are different enhancer or repressor elements which help to regulate the transcription process. Okay, now I will discuss it later bit. Now there is pre-initiation complex formation. Okay, this pre-initiation complex is very much required for formation of the transcription steps, like that of the transcription factor 1, transcription factor 2, then in transcription factor there are different subtypes like the D1, that the A1, that the B1, EH, etc., etc. I will write it here. In the pre-initiation complex, there are different transcription factors. Different transcription factors present. Examples include the TATA binding protein, TATA binding protein, transcription factor 2A, B, E, D, and H, F. Okay, these are different transcription factors which help in the formation of the pre-initiation complex. Okay, without this pre-initiation complex, the initiation of the RNA transcription, DNA transcription will not take place. Okay, now as I said, this D1, this transcription factor 2D helps in the bending of DNA, so that there will be help in the breakdown of DNA sequence. Now the transcription factor 2A and 2B helps in the stabilization of the DNA sequence. The transcription factor helps along with the RNA polymerase 2, and the transcription factor 2H helps in the helicase activity, as the name suggests. So I should write here that the D1 helps in the bending of DNA sequence. This A and B1 helps in the stabilization of DNA sequence. This F1 helps along with RNA polymerase 2, and the H1 helps in the helicase. Okay, now the initiation of the RNA synthesis takes place. Okay, initiation, as I said earlier, the D helps in the bending of DNA sequence. So if I draw here, the DNA sequence is somewhat little bit bent at the point of initiation, and the RNA polymerase is binded here. RNA polymerase. So this one is the upstream, this one is downstream, if I consider it something like that. Now here there will be bubbling formation takes place. And the RNA polymerase will be here, something like this. And as it shifts toward this side, there will be formation of new RNA strand. So the drawing will be something like this one, and the RNA strand forming is this one. New RNA strand. Okay, and it goes on and the strand gets elongated. Okay. And this RNA forms in 5' to 3' direction. Remember this one always. Now the second one is the elongation. Elongation. Here elongation means the new nucleotides bind to the 3' end. Formation of the RNA to 5' to 3' side. There is formation of phosphodiester link. Okay. Now the termination one. Termination. Here also two types can be explained. Two types of termination can be explained by, one is by intrinsic way, another one is the extrinsic way. In intrinsic way there is polyadenylation. And here we can say that cleavage stimulating factor. Okay, now this both will help in the formation of cleavage factor and this cleavage factor, by the help of endonuclease, forms the new RNA. Okay, new RNA is formed. Okay. Now the cleavage symbol is CSF. This is not cerebrospinal fluid, just remember this point. Okay. New RNA is formed after termination. So after this new RNA is formed, there are many modifications that will be taking place in this RNA. So they are known as post-translational modifications. So next point is the post-translational modification. Before post-translational modification, I should show you a diagram here about the transcription. I hope. Okay. Now see here. See, in this transcription process, you see that this is the 5' to 3' strand, and another one is a 3' to 5' strand. We are taking the 3' to 5' strand as the template one, and the another one is the coding, by which we can see here that this RNA which is forming here is same as that of the coding strand. Okay, so the name coding is given, and the transcription process taking place in this direction. So basically this is the transcription process, and here the RNA polymerase is active. I hope this diagram will help you for understanding. Now the next one is the post-transcriptional modification. Okay, so the post-transcriptional modifications, as I said earlier, after the new RNA form, that is known as heterogeneous nuclear RNA, the mRNA has to be formed. So the some transcription modifications include the first one, that is 5' end capping. This capping is done by the help of 7-methylguanosine triphosphate. By 7-methylguanosine triphosphate. Okay. Now this helps in mRNA by giving a stability to the 5' end, and it helps in the transferring of the mRNA from nucleus to the cytoplasm, as I explained earlier also. Second one is the polyadenylation at 3' end. Okay, here if I consider this mRNA at the 3' end, there are different polyadenyl chains present. Polyadenyl chain present. It also helps in the stability at the 3' end. Okay, and third one is the splicing. Now this is an important point which we have to focus. Third one is splicing. Remember this point. In splicing, there is joining of the two exons with each other and removal of the introns. Now what are exons and introns? Exons, I should write here in alphabet. What are exons? Exons are basically the coding sequence which will be coded, and the formation of new proteins will take place. Means coding sequence which will be used for the formation of the protein, and introns are non-coding sequence. So we don't need these introns. Why we will use the introns if it has no function in the coding? So we have to remove this, and the binding of the exons will take place. So the splicing is done by two methods. One is the self-mediated, means self-mRNA is modifying itself for formation of the matured mRNA, self-mediated, and another one is the spliceosome mediated. So these are the two methods. Now in self method, there is no ATP required or no enzymatic action is required. Okay, there are different intrinsic signals which help in the splicing of the mRNA. So if I write these points, no ATP or enzyme required, intrinsic factors present. Okay, Now, in the splices and mediators, there are requirement of spliceosomes. These are enzymes in nature. Require ATP. Now, if I explain this in diagram, let us think that this one is an mRNA, and also for this one is an mRNA, and here the different introns are something like this. Okay. This 5%, this 3%. This has to be spliced. Okay. By the intrinsic factors present, there will be recognition of the sequence, then there will be cut at this point and this point, again this point and this point, and finally cut at these 2 points. They will be cut by different the factors. Okay, so the next will be something like this. Okay, now this formation is taking place, and this spliced one is something present in this loop like way, loop like manner. Finally, this loop is removed and the exons are attached by the help of ligases. So the final formation of the mRNA will be like this, 5' to 3', and mRNA is finally formed. Okay. Now, spliceosomes. If I take this as the 5' end and 3' end, this one are the introns present. Same here, the cut will take here and take here, take here and take here, take here and take here. But this cut will be done by spliceosomes. Okay. After that, these are spliced, and again same loop-like formation is taking place here, something like this. Finally, these loops are removed, and the new mRNA is formed. RNA is formed. Now, along with this kind of changes, there is another change taking place in the mRNA, that is the mRNA editing. We do editing in photos, in videos, same as there is editing is taking place in the mRNA. So the next one is RNA editing. This editing will help in formation of new peptides, new proteins, I would say, from same gene. Okay, means the gene which is present in DNA is the same, but finally the products formed are different in nature. So if I give an example here of the RNA editing, example is apoprotein B. This is an important protein, and this is present in the chylomicrons, HDL, LDL, etc. So in RNA editing, from the same gene, 2 types of proteins are forming. That is, one is B100 and another is B40A. You will remember this from the lipid metabolism. Okay, B100 is present in low density lipoprotein, and it is present in liver, and B40 is present in chylomicron and in intestine. Okay, and this RNA editing takes place by this one. If I take this example, this takes place by editing in the stop codon. While forming the RNA, there is editing in the stop codon, leading to the formation of these 2 types of protein. Editing in stop codon. Now this editing or splicing is very much required, so that the introns are not being used as coding sequence, or else there can be some kind of problems. Generally the introns are removed, but if sometimes the introns are not removed, there will be altered formation of the peptides. Like in example of thalassemia, there is incorrect splicing may be present, or in another disease that is known as SLE, systemic lupus erythematosus, there is improper splicing. So this RNA editing and splicing is very much required. Now the next one is translation, but before translation I should explain you a small topic, that is the genetic codes. So what are genetic codes? Genetic code is the set of 3-letter combinations of nucleotides that is called an each codon, which corresponds to a specific amino acid or stop signal. Like if I give you an example of AUG, okay, this is for methionine, or UCC, this is for serine. But there are some stop codons like the UAG or UAA or UGA. These are for stop codons. You know that there are 20 types of amino acids and 4 types of combinations can be done with these alphabets. So if I make a chart, so 64 types of combination can be seen. Out of 64, the 61 types combinations can represent 20 type of amino acid, and the 3 codons as I explained here, these are the stop codons. So what is genetic code? Genetic code is set of 3-letter, that is nucleotide combination, which corresponds to a specific amino acid or maybe the stop codon. So what are the features? Features is always in triplet form, always in group of 3. The code is non-overlapping. Non-overlapping means they are always present in a sequence. They are not overlapped to each other. Okay. The code is commaless, means there is no gap in the 3 codes. Like if I give an example, A, U, G, U, C, C, U, A, G. Okay, there are no gap present in these points. Okay, it is always present in sequential manner. This has property of degenerate. Degenerate means one codon may have a specificity to different amino acids. This is known as the degeneracy of the genetic code. Means if I take an example of serine. Serine has different codon like UCA, UCG, UCC. Okay, means there is degeneracy present. That means one amino acid has 3 codons present. We need this degeneracy now because there are 20 amino acid and 61 codons present. So there will always be degeneracy present here. Okay, codons. Now there are 3 stop codons present. That are UAG, UAA, and UGA. Now the codons are universal. Means in every organism, AUG always be the methionine. Always methionine. Okay, and AUG is always the start or initiation codon present for any protein formation. Always methionine, and it is the start codon present. Okay. Now the topic is translation. What is translation? Translation is the information present in the mRNA, which is given by the DNA through the mRNA, and this mRNA is translated into the formation of proteins. That means the information present on mRNA in the form of genetic code is utilized for the formation of the proteins. So I should write here, from mRNA to protein synthesis. This process is translation. Okay. Now how the translation process occurs? If I give you an example, like this is an mRNA and these are the different codons present. Okay, so I will see here these 3 codons, 1 2 3 3 3, something like this. These 3 codons will be taken and each will form an amino acid. Okay, and goes on. These amino acids will bind with each other through peptide bond. Okay, now this is a peptide bond. Now during this translation process, all the RNAs are required. That means mRNA is required for the transcriptional process, tRNA is required for the transfer of the proteins, peptide molecules, and rRNA is required so that the peptide bonds can be formed and the protein molecule is made. Means mRNA is for codons, the tRNA is for transfer of amino acids, and rRNA is ribosome. Okay. Now the translation in prokaryotes have different factors. Okay, the different factors for translation in prokaryotes. This includes the initiation factor 1, 2, 3, 4 and 5. Elongation factor has elongation factor G, TU factor, and termination factor has RF 1, 2 and 3. Okay, these are the some factors in translation process of prokaryotes. Now the prokaryotes have ribosomes that is of 7s. Have ribosomes that is the 70s. 70s has two subunits, one is the 50s and smaller one is the 30s. If I make a diagram here, the units will be something like this. Okay, now this one is the larger subunit, that is 50s. This one is the smaller one, that is 30s. Together, when they are bound with each other, it forms the 70s ribosome. Understood? Now the factors in here are also the same steps, that is the initiation, elongation and termination steps. But before these three steps, there is another important step, that is the activation of tRNA. The tRNA has to be loaded with the amino acid. For that, the activation of tRNA is must. Now the other points are initiation, elongation and finally termination. So we will discuss each of the steps. First one is the activation of tRNA. Okay, now in activation of tRNA, there is of two steps. First one is activation of the amino acid. Okay, means the amino acid is activated by the process I am writing in flowchart, like amino acid with ATP forms the enzyme AMP with amino acid. Here the enzyme is the amino acyl tRNA synthetase. Okay, and this one is the activated amino acid. This one is the activated amino acid, and second one is the charging of tRNA. Here this activated amino acid along with tRNA forms the activated or loaded tRNA. So what will be the structure of tRNA? Activated form will be something like this. Excuse me. And here will be the amino acid. Okay, so this is the anticodon arm, this is the D arm, this is the T arm, and this is the variable loop. Okay, now the initiation process will take place. Initiation will take place. That means, first of all, there are two parts of the ribosomes. One, as I already said, one is the smaller, that is 30s, and another one is the larger, that is the 50s. Now this breakdown is done by the help of initiation factor 6. Okay, now the tRNA is formed a little bit different by the help of initiation factor 2. tRNA with initiation factor 2 forms the ternary complex. Okay, so the formation of the whole complex will be the first smaller one subunit along with mRNA along with the ternary complex. Okay, so if I draw these three drawings together, this is the mRNA. If 5' to 3'. This one is something like this, is the 30S rRNA, 30S rRNA, and the complex, ternary complex is something like this. Something like this. Okay, with the amino acid present here. So this is the whole formation of the initiation complex. Initiation complex is formed. Now, after this formation, the larger subunit will come and bind to it. How this will bind? The larger subunit will come, and if I make a diagram here, this is the 5' to 3'. This is the 30S, and above that the larger subunit, something like this, comes and binds to this point, and in which the tRNA gets bind. tRNA gets bind to this point. So the whole 70S initiation complex is formed. This binding of the larger subunit is done by the binding is helped by initiation factor 5. Now this structure has 3 sites. Okay, if I draw this diagram more closely, like this one is the mRNA 5' to 3', and this has a 30S subunit, and this one has a larger subunit. Okay, so here are 3 sites. Okay, one is the A site, one is the P site, and another one is the E site. These are the sites for the tRNA binding or tRNA action. Okay, now the A site is the acceptor site. Here the tRNA comes and binds. tRNA comes and binds. The second one is the P site, means peptide site. Here the main peptide bond formation takes place, means peptide bond formation, or I can say chain elongation. And the last one, that is the E site, is the exit site. So as the shifting of the ribosomes on the mRNA takes place and the chain is elongated, first the tRNA comes to the A site, after that it goes to the P site, it releases the amino acid and the peptide bond is formed, and the remaining tRNA comes to E site and finally goes out of the RNA. Okay, so this is the whole process. Now the next point will be the elongation point. Elongation. As I said, in the elongation, the shifting of ribosome take place over the mRNA, and peptide bonds will form. The first amino acid to be binding at the A site is the methionine. Okay, the first amino acid will be methionine. After that, different amino acids will be coming and the peptide bond will form. Okay, now the final stage is the termination one. That is termination. Now in termination, as I said earlier, in the genetic code part there are 3 stop codons. The 3 stop codons are UAA, UAG and UGA. So if I make a diagram again here, like this is the mRNA 5' to 3', and here I can see the 3 codons. For example, UGA. Okay, so the tRNA, no tRNAs are present for this thing. Okay, so when this come, and here is a tRNA already present, as I said. Okay, and the final amino acid is binded here, and the other peptide forms like something like this, and it goes on prolonged. So finally this one is the peptide bond, and these are the stop codon. So finally the termination takes place when these codons come. Okay, and different factors also take place in the termination process. So stop codons are stop codons are UAA, UGA and UAG. These are the 3 stop codons, and the final termination takes place. Okay, and finally peptide chain is made. Now the translation in eukaryotes a little bit different from this prokaryotic one. Here also maximum will be same like that of prokaryotes, like the activation of the tRNA. In tRNA, the amino acid with ATP forms the enzyme AMP and activated amino acid complex, then activated tRNA complex, then again there is initiation. But here is the difference in the process, that is the change in translation factors, as translation factors are different in prokaryotes and the translation factors are also different in the eukaryotes, because in eukaryotes and prokaryotes there are always some kind of difference. Obviously there it will be present. Okay, so the translation factors in eukaryotes are for the initiation factors. For initiation are the eukaryotic initiation factor 1, 2, 3. Then there will be initiation factors of 5, 6, 4. Okay. Second one, elongation. These are easy to remember. I hope that you will remember all these points. It is not required too much, but if you can remember, then it will be a age point for getting good marks. EEF 1 alpha, then EEF 2, and finally the termination. That is the ERF1, ERF3. So these are the some translation factors in eukaryotes. Now the some are other differences in the eukaryotic and prokaryotic translations are, I should make a table here. Eukaryotic, prokaryotic. Here in eukaryotic the first amino acid is the methionine, but in case of prokaryotic it is formyl methionine. Many translation factors. Less factors. The ribosomes here are the 80S and here are the 70S. Subunits include the 60S plus 40S, and here include the 50S and 30S. Okay. And the elongation and termination factors are also different. Okay. Elongation and termination factors are different between the eukaryotic and prokaryotic. So these are the some differences between the eukaryotic and prokaryotic translation. Now the inhibitors in the translational process. This is very important. Inhibitors in translation. I can divide it in 3 parts. That is, one just for prokaryote, one just for eukaryotes, and last one is for both. For prokaryotes, some examples are tetracycline, erythromycin, neomycin, and for eukaryotes it is lysine, cycloheximide. For both it can be puromycin. Okay, so these are the some points. Now if I mark the important points from the starting, I have missed the important points in the genetic codes. So genetic codes are 3-letter nucleotide for specific amino acid, always in triplet form, non-overlapping, homologous nature and degeneracy in nature. Stop codons are these 3, and starting codon is always AUG. Translation, there are 3 parts. Okay, the factors for translation in prokaryotes, sorry, these are the points. Now the steps are these 4: activation of tRNA, initiation, elongation and termination. Now in activation of tRNA there is activation of amino acid and charging of tRNA takes place. Then after that initiation takes place. In initiation there is initiation complex formation, and this initiation complex has 3 sites, A, P and E. And in elongation there is shifting of ribosomes takes place and the peptide bond forms. The first amino acid will be methionine. In termination, these top codons are these three, and finally the peptide chain is formed. Now the translation factors in eukaryotes are these. Okay. The difference you should remember between the eukaryotic and prokaryotic are the methionine and formal methionine, many translational factors and less translational factors, and you can write the specific translation factors difference between the eukaryotic and prokaryotic as I already given example in this point. And elongation and termination factors are also different between the eukaryotic and prokaryotic. And finally, the inhibitors of translation for prokaryote, the eukaryote, and both are these examples. Okay. Now the post-translational modifications. Post-translational modifications. The post-translational modifications can be explained by two types. One is the covalent modification and another one is the proteolytic cleavage. Okay. Covalent modification and another one is the proteolytic cleavage. Okay. In covalent modification, there are different points like the glycosylation, disulfide bond, gamma carboxylation, acetylation, phosphorylation. These are some examples of covalent modification. In proteolytic cleavage, there can be removal of methionine or proteolysis between the chains, and this can be seen in the I would say in insulin. Here there are three subtypes A, B and C, and the C is removed. Okay. And other can be proteolysis in the preformed chain. Okay. So these are some kind of post-translational modifications. Now I will give example for each of these. Okay. Like glycosylation. Glycosylation is the attachment of carbohydrates so that the active protein is formed. Glycosylation, as the name is already suggesting. Disulfide bond between two cysteine residues. The sulfide bond is generally formed. Okay, the cysteine has sulfur present. Thus, the sulfide bond is formed. Third one is gamma carboxylation. Gamma carboxylation is seen in the vitamin K and activation of the clotting factors. Acetylation, addition of the acetyl group, and phosphorylation is activation and deactivation of enzymes basis on the whether the protein is phosphorylated or not. These are the some post-translational modifications. The topic is lac operon. Now, what is operon? Operon is a set of genes which is involved in a single function. So the function mainly is composed of the metabolic pathway. Okay. So what is operon? It is a group or set of consecutive genes in a same strand in a genome that has co-transcribed into a single polycistronic message. Okay. Now here polycistron means many proteins. Okay, many proteins from a single mRNA. So this is the basically operon concept. This concept was given by Jacob and Monod. Concept was given by Jacob and Monod. Now here the lac means lactose. Thus the lac operon we are discussing here is the metabolism related to the lactose in a lactose tolerating bacteria. So lac operon means lactose metabolism regulation in an organism, generally bacteria. This operon concept is mainly present in the prokaryotes. Okay. Now if I should show you a diagram here. if you see this diagram here, this is basically the lac operon concept. Okay, now this is the lac operon. Here we can see the lac I. Lac I means inhibitor, means inhibitory protein formed. Okay, now second is the P zone. This is the P zone, where P means promoter, and promoter means there is binding of the RNA polymerase. Binding of RNA polymerase. And lac Z means there is transcription then translation of different proteins. If I write it like this, lac Z means the beta galactosidase. Lac Y means there is translation of the galactoside permease. And lastly, lac A means the transacetylase. So these are the main points in the lac operon. Now, if I see this lac operon here, what is the concept? First of all, if I consider three situations. First, when the glucose is available. If glucose is available in the medium, then the bacteria don't have to metabolize the galactose or the lactose. So this operon system will be repressed. Okay. Now, second, if glucose and lactose both are present, then also it will be repressed. Why? Because there is glucose present. So why we will take the lactose? Because its metabolism is quite difficult, and this whole concept has to be done in the cell. And the third one is if only lactose is present, and if we see that if only the lactose is present, then the bacteria will take up the lactose for its metabolism. Okay, now the lac operon action will take place. Okay, so what is the concept? Lac I means the inhibitor. Inhibitor means inhibitory protein. Lac I have the inhibitory protein. This inhibitory protein always inhibits the RNA polymerase to bind in the promoter's region. Okay, but if I should write this function, inhibits the binding of RNA polymerase to the P site, okay, or the promoter site. So if the protein is binded to the P site, then the whole translation will not take place, and these enzymes will not form. But if it is inhibited, if the protein is inhibited, then the whole translation process will take place and the proteins will be formed, or I would say the enzymes will be formed. So the inhibitors of this inhibitory protein are lactose itself and allolactose. As here it is given also lactose. See, if only the lactose is present, then we have to induce this operon system in the bacterial system, and the protein inhibition is must required. For this protein inhibition, these induced protein inhibitors, that is the lactose and allolactose, is required. And in the surroundings, if only lactose and allolactose present, the cell will take this lactose, the protein is inhibited, and finally the translation takes place by the transcription takes place, and finally the mRNA formed, and then the proteins are formed as required for the metabolism of lactose. So this is the basic concept of lac operon, and each functions, that is the beta galactosidase, has function of glucose plus galactose from lactose. This is the function of beta galactosidase. Okay. Now the second one is the permease. This has function of permeability regarding the lactose entry in the cell. If the permease is present, then the lactose entry will take place. Lactose entry. Okay. And the last one, that is transacetylase. Now here we know that acetylation is a secondary conjugation function reaction. So there is modification after the metabolism of lactose to the metabolites, so that there is detoxification, or else the metabolites which are forming will be toxic for the cell. So mainly detoxification action by conjugation. Okay, so these are the function of the beta galactosidase, permease and transacetylase. Okay, and if after some time there is glucose present in the medium, again there is in decrease in the lactose absorption or permeability, thus the allolactose or the lactose is decreased, and finally the inhibitory protein is its action will be resumed again, and after resuming again the transcription and translation will be stopped, and these enzymes will not be formed again. So this is the basic concept of the operon concept. Operon concept. I should write these 3 points here, the 3 situations I have given, like if glucose present, then lack operon, not any action, or I would say repressed. If both glucose and lactose present, again repressed, and if only lactose present, then lack operon induced. So these are the 3 situations we are getting, and after that the lack operon action we are seeing. Okay, now the next topic is the recombinant DNA technology. Recombinant DNA technology. So what is recombinant DNA technology? It is the recombination of different genes or different parts of DNA to the for forming a new DNA product, and this DNA product is helpful for the human beings. Okay, so it means the DNA we are finally forming has a desired product in our lives. So what is recombinant DNA technology? It is basically recombination of desired DNA segments with other segments so that we can get desired products. Okay. So there are different steps in the recombinant DNA technology. Okay, and different enzymes and different processes are taking place in this technology. So what are the steps basically? First step, so steps of RDT. First step is the isolation of the desired DNA. You know, we require the desired DNA for its fully functionality and usage in our lives. If we don't get the desired DNA, how we will get recombination done with the other DNA so that the fruitful result will get? So the isolation of the desired DNA is required. Then isolation of vector. We require a vector or a carrier so that the desired DNA is to be taken to the host cell and the further replication and its action will take place so that the desired products we can get. So isolation of the vector. Formation of hybrid, or I would say recombinant DNA. Okay, along with the vector and the desired DNA, the recombinant DNA is formed. Then introduction into the host cell, then multiplication in the host cell, and finally getting desired products. So these are the basic steps of the recombinant DNA technology. So and now there are different enzymes we are using for its action. Okay, and different vectors we are using. So I would write these points, then after I will discuss each and every step of the recombinant DNA technology. So enzymes requiring are the restriction endonuclease, nucleases, I would say. Okay, or I would say restriction endonuclease. So what is restriction endonuclease? Endonuclease is basically a molecular scissor. It's act on the DNA strands and cuts the desired part of the DNA. Okay, nucleases are basically of two types: exonuclease and endonuclease. Exonuclease means it acts on the terminal nucleotides, and endonuclease acts in between the strands. Okay, so endonuclease means in between the strands they are acting. Now the second one is the DNA ligase. Ligase means ligation of the DNA with the vector DNA. Okay. Then the DNA are the DNA polymerase for the replication of the desired or the recombinant DNA. Reverse transcriptase may be required for the changing of the DNA, for the changing of desired RNA product into the DNA product. So these are the basically enzymes which we are requiring. Now each and every step I will be discussing here. So the first step is isolation of the desired DNA. Isolation of desired DNA. Okay, here we take a cell of our interest, then we use enzyme detergent for its removal of the overall coating of the cell. Then after that we use the phenol or phenol protease and ribonuclease for the removal of other components of the cell, the proteinaceous components, the carbohydrate components, and the ribonuclear components, because we need only the DNA. Then after that we do the ultracentrifugation, and after that we use chilled ethanol, and the DNA strands are precipitated out. So I should write the steps here. That is the cell taking. After that, enzyme detergent used, action of the phenol, ribonuclease, and the protease is used. After that, ultracentrifugation, chilled ethanol, and finally DNA precipitated out. Now this precipitated DNA is done electrophoresis. Why electrophoresis is done? This step is very much required for the identification of the desired DNA. Identification of the desired DNA. Then finally the desired DNA with sticky ends. These sticky ends are the extra two-sided ends which are required for the adhering of the vector DNA with this desired DNA. So what are the steps we have discussed here? That the cell taken, enzyme detergent used, in which the phenol, ribonuclease, proteases, etc. used, ultracentrifugation, after the chilled ethanol, DNA precipitate out. After that electrophoresis is done for getting the desired DNA. Okay, this step is required for taking out the desired DNA. Okay. Now second step is the isolation of the vector. Isolation of vector. Now this vector is very much required for the attachment of the desired DNA and transferring it to the host cell. Without the vector, the desired DNA can't be taken by the host cell, and the desired DNA will die off or it will be destroyed. Generally the vectors are of different types. Now vectors are of different types. I should first explain you the types of vectors. So types of vector and their properties. Okay, so what are the different vectors here? The different vectors like the plasmid. Okay, now next is the plasmid has special characteristics is that it is extra chromosomally present in the bacteria, and it is double stranded, and another special feature is that it can replicate by itself without any connection to the main bacterial chromosome. So what are the characteristics? Extra chromosomal, can divide itself, and last one is double stranded. Second one is the cosmid. So what is plasmid? Plasmid with bacteriophage. Okay. Now the third one is bacteriophage. You know that bacteriophage can also transfer DNA. Okay. By taking the bacterial DNA after replication in that bacteria, the bacterial DNA is taken by the newly formed bacteriophage and it transmits to the other bacteria they are infecting. Bacteriophage can be a medium. They are viruses. The other ones are the yak, that is yeast artificial chromosome. Okay, this yeast is helpful for cloning of the larger DNA segments. For large DNA segments. Next is BAC, B-A-C, bacterial artificial chromosome. Okay, so it uses other bacteria for the transferring of the desired DNA. Other bacteria use. And the last one, that is MAC, M-A-C, that is mammalian artificial chromosome. It is also for large DNA segment. So these are some kinds of vectors. Now, as we were discussing, isolation of the vector. In isolation of vector, if we take a plasmid, then the plasmid we have to isolate. So we have to first destroy the cell of that bacteria. How we can destroy? By using the lysozyme enzymes. So after using the lysozyme enzymes in the presence of EDTA, the cell wall is destroyed. Then with the help of ultracentrifugation, the plasmids are isolated, and with the help of electrophoresis, the desired plasmids are also isolated. So we take this plasmid, and with the help of restriction endonuclease, we cut the part of the plasmid. So we get plasmids again with sticky ends. So I should write here the points. That is plasmid with cell wall. We destroy it, cell destroyed with lysozymes. Now, after destruction, the inner contents of cell are present. We take this and ultracentrifugation is done. After ultracentrifugation, the different plasmids are obtained. Okay, we do the electrophoresis and the desired plasmid is obtained. Again, we use the restriction. Why the restriction is given along with the endonuclease name? Because we are restrictionally cutting the plasmid or the bacterial DNA. We are not cutting here and there and any point we are thinking of. We are cutting in the restricted points. So restriction endonuclease. Restriction endonuclease. And finally, plasmid with sticky end. Generally, ends are of two types. One is the sticky one and another is blunt end. Okay, I will discuss about the sticky and the blunt end in later bit. Okay, so these are the points for the isolation of the vector. So I should say again, what are these points? Plasmid, cell is destroyed. After that, inner contents of cell are taken. Ultracentrifugation done. Different plasmids are obtained. Electrophoresis is occurring. Desired plasmid is obtained. By the help of restriction endonuclease, plasmid with sticky ends we are getting. Now, there is formation of the hybrid DNA. Formation of recombinant DNA. So, if I draw a diagram here, like think that this is a plasmid. This is a plasmid and a part of it is cut. Okay, so this is the restriction. Okay, this is the plasmid and the desired here. If we see that a little bit is getting out here, these are the sticky ends. Sticky end. And along with it, the extra part which we get from the desired DNA, that is desired DNA. So it finally forms the recombinant DNA. So what it will look like? It will look like something like this. So we are getting this one. Okay. So this is the recombinant DNA. This adhering is taking place by the action of DNA ligase. DNA ligase. Now this recombinant DNA has to be transported into the host cell. Okay. The fourth one is the, here we have explained, is introduction into the host cell. Into the host cell. So we can transfer this DNA by both natural methods or the artificial methods. So I will classify it into two parts. One is natural and another one is the artificial methods. So in natural methods, the different natural methods can be conjugation, transposition, bacteriophage action, retroviral transduction, and agrobacterium mediated transfer. Okay, and the artificial methods include the microinjection, protoplast fusion, heat and cold action, means repeated heat and cold changing of the environment along the bacteria, so that there will be increasing permeability and they will take the recombinant DNA. And ballistic gun use. Okay. So these are some of the processes. Microinjection means there is microinjection present in which the needle is too much thin so that it can penetrate a cell, a single cell. The microinjection is taken and the recombinant DNA is taken and it is injected in the cell. Now the next one is the protoplast fusion. As the name suggests, two different protoplasts are fused and there will be exchange of the recombinant DNA. Heat and cold action, as I have already discussed, and ballistic gun. In ballistic gun, tungsten or gold is used and the recombinant DNA is coated with these two metals and it is shoot to the cell, and after shooting it gets penetrated into the cell. Like normally we use gun and the bullets penetrate into the body, similarly this ballistic gun is used and the recombinant DNA coated with gold and tungsten and shoot into the cell so that it will take the recombinant DNA. Now the fifth one is the multiplication in the host cell. Multiplication into the host cell. There we will choose those kind of cells which have less antibiotic resistance, or I would say specific antibiotic resistance. Okay, or else if we give the DNA and if we provide it with different kind of antibiotics, the bacteria may die. So we will choose a specific kind of bacteria for its multiplication. Now the last one is formation of desired product. This desired product is helpful in our day-to-day lives. Okay. Now, as I have explained this, now I will explain a specific type of plasmid. So I should draw a diagram here. Okay. Now, this is known as PBr322. Okay. Here P means plasmid, Br means the founders of this plasmid, that is Bolivar and Rodriguez. and 322 is the number of the plasmid. Okay, now it has different components, like one which we are explaining here is something like this. This is known as BAM-H1. H1. This gene is known as BAM-H1. This is required as tetracycline resistant gene. With presence of this gene, the bacteria will have tetracycline resistance. Another one present here is the PST1. It gives ampicillin resistance. Okay, now here another is present. This is known as the ORI site, means origin site. So it is a plasmid with two antibiotic resistance genes. The antibiotics are the tetracycline and another one is ampicillin. Okay, generally this plasmid is present in the E. coli, okay, and in eukaryotes, in some fungi and plants. Okay, present in E. coli, some fungi and some plant cells. These are some examples. Now the important points here are the tetracycline and ampicillin resistant, and the specific name for this plasmid is PBR322. Okay, now the applications of RDT. First one is insulin formation. In earlier times, the insulin injections were made from the bovine or the insulin from the pigs. So these insulin have allergic reactions on the different patients. So after the recombinant DNA technology, the newly formed insulins are not providing any kind of allergic reactions in the host, or I would say the patients. So it is very much helpful for the insulin formation. Like I would draw a diagram here. Okay, for this is the bacteria and this is another bacteria. Okay, or I would say these are two plasmids. In this plasmid P1 and P2, in this plasmid the gene for, you know that the insulin has 3 chains. That is, if I draw a diagram like this one is A, this one is A, B and C, and 2 sulfide bonds are formed here. Okay, sulfide bonds are formed here, and this C is removed so that A and B forms the main insulin enzyme. Okay, we can do now that we insert a gene for A chain and we insert a gene for B chain. Okay, so after introduction to the host, this DNA are expressed and the A chain and the B chain are formed. A chain and B chain formed. Okay, now these both chain are purified. After purification, they are attached with each other by bisulfide bond. Each other by 2 disulfide bond, and the new insulin is formed. So the insulin structure will be something like this. I should draw it here much more better way. This one is A chain, this one is B chain, and here 2 sulfur bonds are present, something like this. So this is the mature insulin structure. Now other examples are different vaccines, which include the hepatitis B vaccine, and some other vaccines. Okay, DNA vaccines, I would say. Other DNA vaccines. Other DNA vaccines. It can be used for production of hormones like the FSH, LH, GH. Okay, like the GH, FSH, LH. Okay. Now it can be used, which was used extensively in Human Genome Project. It can be used in DNA fingerprinting, gene therapy, and there are different other examples. These are the main examples in which the RDT is done. Now another topic is PCR. PCR. PCR stands for Polymerase Chain Reaction. Okay. This chain reaction is done so that multiple copies of the specific DNA can be obtained. Means after using the DNA polymerase, there is chain reaction, means exponential growth reaction, so that many copies can be obtained for the research purpose or the investigation purpose. So as the name suggests, use of polymerase chain means many products, many, I would say, copies for research or investigation work. Okay, so this is required by the action of the PCR. So what are the requirements in this PCR technique? There is a template DNA or the desired DNA, I would say. Desired DNA is required. PCR machine is required. PCR machine is required. This PCR machine is also known as thermal cycler. Okay. Now different primers are also required. You know that for replication process, RNA primer is a must required thing. So different primers are also required. Specific polymerase, specific DNA polymerase, that is the TAC polymerase is required. TAC means Thermus aquaticus. Why it is required, I will tell it later. TAC polymerase and different nucleotides are required. So the process of PCR is done in 3 steps. Steps of PCR. Okay. So what are the steps of PCR? First one is denaturation. Denaturation. In this step, the separation of the double strands takes place. This is done in the high temperature. For approximate high temperature is 90 to 95 degrees Celsius for approximate 1 minute. So separation of strands in high temperature, that is 90 to 95 degree Celsius for approx 1 minute. Now the second one is annealing. Okay, in annealing, renaturation takes place. Why? So that attachment of the RNA primer takes place at temperature cooled down to 56 degrees Celsius for 30 to 40 seconds. Now the third one is extension. Now this is an important point in the PCR process. So synthesis of new DNA strands at temperature of 72 degrees Celsius. Okay, so time taken approximately 2 to 3 minutes and cycles taken is 20 to 25. Okay, this time is taken for each cycle, and number of cycles, total cycles taken is 20 to 25 times. So if I make a diagram here, so if I draw a diagram here, this is a DNA. Now these are the phases. First is the denaturation. Denaturation taking place. Two template strands are formed. Two template strands. Okay. Now, annealing takes place, and primers are added. So what will happen here? One is this, another one is this. So here a small primer is attached. Primer is attached. Now denaturation has temperature of 90 to 95 degrees Celsius, and annealing has temperature of 56 degrees Celsius. After this, there is finally next step, extension. Extension means two DNA strands are extended and new DNA is formed. Okay. New DNA strand. This extension takes place at 72 degrees Celsius. Okay. Remember these temperatures. These temperatures are asked in Viva or in MCQs. Okay. So approximately 25 to 30 cycles are done, and about a million copies can be obtained. Why? There is exponential growth of the DNA strands. So exponential growth of DNA replication. So about a million copies can be obtained. So what are the applications of PCR? Now the applications of PCR are the SARS-CoV-2 detection. SARS-CoV-2 detection has recently occurred. Other viral infections, detection of cancer, in recombinant DNA technology, in forensic medicine, and RT-PCR. RT-PCR means real-time PCR, means checking of viral load. Okay. Now in RT-PCR there is checking of the viral load. Means while checking of the viral load, if I say like we need a specific quantity of viral content, if more cycles are required for getting that amount of viral content, means the viral load is less, and if less cycles are required to get that specific amount of viral content, that means the viral load is more. So I have explained it here, the connection between the cycles of PCR and the viral load. Okay, and these cycles are calculated at that instantaneous time while the PCR is taking place. Thus the name RT-PCR is given, means real-time PCR. Other types include the Tachman probe. Tachman probe or SYBR gene. This is done by the help of the fluorescence amount. I have missed one part. I should write here. The Tach polymerase. Tach polymerase. This polymerase is obtained from Thermus aquaticus. This organism is present in high temperature water, means in spring hot waters, and their polymerase enzyme does not degrade in high temperature. So it will be helpful in high temperature like 72 degree and 80 degrees Celsius for the proliferation and extension of the chain. So obtained from Thermus aquaticus, and special features include the polymerase acts in high temperature. I have missed this part. So as I was explaining here, the Tachman probe or the SYBR green. Now this is a type of fluorescent material in which the fluorescence amount is calculated. By the help of this fluorescence amount, we can detect the amount of the viral load or any kind of viral content present. So if I write here, more viral load means more fluorescence, and less viral load, less fluorescence. Okay. Now here is a trick that the Tachman probe has two parts. One is the fluorescence part and the reporter of the fluorescence part. Okay. Now when the DNA synthesis is occurring, the fluorometer detects the change in the fluorescence amount. Okay. If the viral load is more, the fluorescence amount will getting more and more and more. So this viral load, for example, if I take the example of COVID, okay, if if I show it in a graph, something like this, okay, we know that the viral load has a threshold point after which we can say that there is viral infection. Viral infection threshold, and we are seeing it, we are doing the cycles and cycles, and the fluorescence amount is increasing somewhat like this. Okay, somewhat like this. This is viral load and cycles. Suddenly we see here there is spurt in the growth of the viral load. That means the viral infection is present, okay, after crossing the threshold level. So in this way we can check the whether there is viral load present or not, or means viral infection is present or not. This graph is done by the help of fluorometer. I should show you a diagram here about the recombinant DNA technology. See here. As I was explaining, in the recombinant DNA technology, there is cut or split of the plasmid part, the specific enzyme. Specific enzyme is the restriction in the nucleus. Then there is selection of the desired piece of DNA, cut and inserted into the DNA, and finally the recombinant DNA is obtained and it is inserted into the host cell. I got this diagram from internet. You can see this. I thought of sharing this to you, so I gave this diagram to you. RDT diagram. Okay. Now the next topic is the mutation. Mutation. What is mutation? Mutation is the permanent alteration of the nucleotide sequence of a genome of an organism or any other genetic elements. I should write here. A mutation is the permanent alteration of nucleotide sequence of the genome of an organism. It may be genome or extra chromosomal DNA or any other genetic elements. May be DNA or RNA or extra chromosomal DNA. Now, what are the causes of mutation? The mutation can cause due to mainly two reasons. One, mistakes in the cell replication, and another one is the environmental factors. Main causes include mistakes in the cell replication or environmental factors. Now, there are different types of mutations present. So, different types of mutations are mutations. It is also divided into two parts. One is the chromosomal mutation and another one is the gene mutation. Okay. Now, under chromosomal mutation, there are four types of mutation. So, what are the four types? The four types are: first one, that is deletion; second one, duplication; third one, translocation; and the last one, that is the inversion. In gene mutation, there are also two types. One is the point mutation and another one is the frame shift mutation. Under the point mutation, there are three types. One is the silent, another is missense, and last one is nonsense. Under frame shift mutation, there are two types. One is insertion and another one is deletion. Now we will discuss this one by one. First one is the chromosomal mutation. Mutation. In chromosomal mutation, there can be some kind of missing or aberration or addition of extra parts in the chromosome. This can be done while the cell replication process, and the process may forms the atypical number of chromosomes or the structural abnormality in one or more chromosomes. So I should write this point: any aberration or addition, deletion, or extra attachment in chromosomal DNA. So this leads to atypical number of chromosomes or may have structural abnormality. Now the first one is the deletion. So what is deletion? Deletion occurs when the group of nucleotides are left out in a gene or excised out in a gene. They usually cause a shift in the reading frame that will finally truncate the formation of the proteins. Now the deletions can be caused by the errors in the chromosomal crossovers during the meiosis process, which can cause different types of diseases. Now I am writing these points. That deletion occurs when nucleotide are left out or excised out from gene or genes. Okay, this will lead to removal of some part or parts of genes. Thus abnormal or no proteins may form. Okay. And deletion may lead to deletion occurs during the chromosomal crossovers. During meiosis. This lead to several serious diseases. Some examples include the Cry-do-Chat syndrome, and other examples are the Prader-Willi syndrome. Now if I show this with a diagram, like for example, this is a chromosome. Okay, these are different parts. Now while crossing over, this part, if I draw with another color, this part gets deleted. So the new one will be something like this. This one is normal and this one is little bit somewhat smaller. So there is some aberration present, abnormal chromosome. Now, second one is the duplication. As I said here, duplication. As the name suggests, duplication means a portion of chromosome will be duplicated and it will be repeated. Okay, leading to extra lengthy chromosome. Means a part of chromosome is duplicated, resulting in extra genes, or I would say genetic material. This occurs when there is unequal crossing over during meiosis. Okay. And this unequal crossing over during meiosis takes place in the homologous chromosomes, or else the same gene will not be repeated. If two other different chromosomes have taking place the crossing over, then the duplication will not be present, may or may not be present. But between the homologous chromosomes, it is always seen that if there is some kind of problem, it can lead to duplication. Homologous chromosomes. So if I draw a diagram here, this one is a normal chromosome and this one is another homologous chromosome here. And if crossing over takes place, and if there is some kind of problem regarding the crossing over, so the duplication will take place. Thus the newly found will be something like this. I will point out the duplicated or the aberrant region with a different color. another one is, so here are one. So here we can see that if I mark out these points as 1 2 3 4 5 6 7 8, again here 1 2 3 4 5 6 7 and 8, after crossing over I can see that 1 2 3 4 5 6, again 5 6 is here, or I would say 7 8 and 7 8 is present, and 1 2 3 4 and 5 6. So here this point is getting duplicated. So this is duplicated chromosomal part. Now the next one is inversion. Now what is inversion? Inversion is the portion of a chromosome that is broken, turned upside down and then reattached. Thus the genetic material is inverted. So what is inversion? As the name suggests, portion of chromosome is inverted or turned upside down and then attached. Thus the genetic material is inverted. So if I show this with a diagram, like now this is a chromosome, and after there are some parts 1 2 3 4 and 5. This one is 1 2 3 4 and 5, but some kind of mutation leading to inversion. Here I can see that 1 2 3 4 5, and I should draw it more clearly. 1 2, now 5 4 and 3. So the problem here is present in this point. Here I see that there is presence of inverted parts of the gene, that is 5 4 and 3. So inverted genetic material means inversion. And finally the translocation. What is translocation? Translocation is basically rearrangements of different parts of chromosome with the non-homologous chromosomes. So what is translocation? Translocation means between non-homologous chromosomes there are exchange of genetic materials. If I show this with a diagram, like this one is a chromosome, and here another chromosome present. Here. Now here we can see that there is translocation between this one and this one. So there will be some kind of change between the non-homologous chromosomes. Finally, the formation will be something like this. So this is the formation of the two types of chromosomes. New chromosome formation. Okay, so this is the translocation. So if I again mark these points, what is deletion? When nucleotides are left out or excised out from the genes, okay, lead to removal of some part of the genes. It occurs during the chromosomal crossovers. Examples include the Friedreich syndrome, Prader-Willi syndrome. In deletion, the part of chromosome is duplicated, resulting in extra genes and extra genetic material. Unequal crossing over between meiosis in homologous chromosomes, as I already shown here. Inversion, portion of chromosome is inverted. And translocation is non-homologous chromosomes exchange of genetic material by crossing over. Okay. Now the gene mutations. So gene mutations. What is gene mutation? Gene mutation is a permanent alteration in the DNA sequence that makes a gene such that the sequence differs from what is found generally. Okay. Now the mutations can range in size. It can affect from one nitrogenous base to one nucleotide, or it can be of many nucleotides, or it can be of multiple genes. Okay. So right here, permanent alteration in DNA sequence that sequence differs for normal ones. Now this mutation can range from mutation size can range from one nitrogenous base to one nucleotide and up to multiple genes. Okay, so this is the definition of gene mutation. Now it has different types of gene mutation. One is the point mutation, and as I explained here already, that one is point mutation, one is frame shift mutation. Now we are explaining the point mutation. So what is point mutation? Point mutation is a type of mutation in which a single nucleotide base is changed. This change can be insertion or deletion of the genetic material. Okay. So point mutation. Point mutation is a type mutation that causes a single base pair, or I would say nucleotide. There can be substitution or insertion or deletion of genetic material. Now this point mutation is of basically three types. One is the, I will write here, point mutation, and it has three types. One is the silent, one is nonsense, and another is missense. Okay, so now the first one is silent. Silent mutation here means, if I take an example, like in mRNA level, for the lysine, the genetic codon is AAG. But if there is point mutation such that in place of G there is A is present, means AAA. So the AAA also codes for lysine. That means even after mutation, the lysine is present in the protein. That means there is, even after mutation, no morphological changes is seen in the peptide chain. So this is the silent one. Example, no change seen. Okay. In nonsense point mutation, there is a change to a stop codon. The stop codon are the UAA, UGA, and UAG. If there is point mutation and there is formation of one of these three codon, then it will be nonsense mutation. And finally the missense, change of one type of codon to another type. Okay, so these are the three types of point mutation. Now if I give some examples here of the lysine. Okay, example, AAG means lysine. In silent mutation, if it changes to AA, also lysine present. Okay, but if in nonsense, in place of AAG, UAG is formed, means stop codon. Thus mutation lead to stop codon. And in missense, if AAG is lysine and in place of second A ACG is formed, and ACG is threonine. So another amino acid is present in place of lysine. So these are the point mutations. I am pointing this one: silent, nonsense, missense. No change is seen. Change to a stop codon and change from one type of codon to the another type. Okay, and the examples are given here. Now the next one is the frame shift mutation. Now, what is frame shift mutation? As the name suggests, frame shift, there is shifting of the codons in the DNA sequence. Now, this shifting can be done by two processes. One is the deletion of some nucleotides, another one is insertion of some nucleotides. Thus, the shifting of the frame of the DNA can be done. So, shifting in the nucleotide framework. Shifting in the nucleotide framework so that the DNA sequence is altered. Now it has two types. One is deletion, another one is addition, or I would say insertion. Okay, now in deletion there is deletion of some nucleotides. Okay, deletion of some nucleotides. Example, if I give like ACCG, TACG, okay, T. Now here I can see that there are three codons present. Now if I delete these two, this T and this A, so what will be the net formed? ACCG CGT. Means there is change in the framework of the genetic code and the new forming amino acids will be different. Like here you see that GTA is present and CGT is present, but now here we can see that ACC is present, but it is changed to CGC and the further one are also present. Okay. Now in insertion, there is insertion of some nucleotides. Like if I give an example, similarly ACGTACTTACG. Now here I can see that these are the codons. Now I see that two codons are added here and here. So what will be the net formed? Here G is added and here A is added. So what will be the new codon? ACGTACGTTAACGT. Okay. So this one is present. This one is present. Now here see that it is changed to GTT. First TTA was present, but it is now changed to GTT, and finally here you see that CGT is present, but now it is changed to AAC, and it goes on. So there is change in the amino acid formation. So these are the two types of mutations present under the frame shift mutation. Now some other mutations include the complete change in the chromosomal number. Some other mutations include the change in the chromosome number that occurs during the improper disjunction, that is the non-disjunctions of chromosome. During non-disjunction. Okay. Some examples include the Down syndrome or the Turner syndrome or Klinefelter syndrome. Okay. Now I should write examples of the diseases that occur due to this type of mutation. First, under the chromosomal mutation, maybe I should use another color. Under chromosomal mutation, there is deletion, duplication, inversion, and translocation. Under gene mutation, one is silent. Silent has no disease, so I am removing this point. Missense, nonsense, insertion, and finally deletion. Okay. Under deletion of the chromosomal mutation, there is Ribosomal syndrome or the Prader-Willi syndrome. Under duplication, some are Smith-Magenis syndrome. Another is Williams-Buren syndrome. These examples are given so that when they are asked about the mutations, you can give the examples in Viva or in MCQs. You don't have to remember about each and every disease. It is difficult for you to understand all these diseases now. Okay. Now the inversion. Hemophilia. Another is Hunter syndrome. Under translocation, it will be leukemia. Under gene mutation in xylem, there will be no disease present. In missense, under missense it will be sickle cell anemia. Under sickle cell anemia, and other will be epidermolysis bullosa. Epidermolysis bullosa. Under nonsense, it will be cystic fibrosis or some kind of metabolic disorders. Okay. Under insertion, some examples are Fragile X syndrome or myotonic dystrophy. And under deletion, there are some examples like DiGeorgi syndrome. So these are the main points. You should mark these important points. In under frameship, nucleotide framework is shifting, under which there are two types. One is deletion, one is addition or insertion. Deletion of some nucleotides and insertion of some nucleotides. Other mutations include the change in the chromosome number, like that in case of Down syndrome, in Turner syndrome or Klinefelter syndrome. Now the examples of each and every mutation is given here. I hope that you will understand, that you will all not understand these points, because these are important. These are a little bit tough for you to understand, and after some time, after first year or second year, you will slowly know about each and every diseases. So thank you very much.